much faster than that.
Well, a new study may have found the reason why a treatment for Alzheimer's disease is having an adverse effect on humans not seen during the clinical trials. Joining me now to explain is Dr. Stuart Lipton from Scripps Research. Doctor, good to talk with you tonight.
Thank you for having me on, Ginger. Good to hear you.
Well, I read the study. It's compelling. This is really fascinating information, including kind of the unexpected find that we'll get to. But first, for our viewers, identify what the problem was that you were addressing.
Well, we don't have a really good treatment for Alzheimer's. The latest treatment came out over 10 years ago. It actually came out from my lab when it was in Boston. We need better drugs for both Alzheimer's and Parkinson's disease. One approach is to been developed antibodies to remove the proteins that are misfolded. In the case of Alzheimer's disease, the primary one is called beta amyloid, and Parkinson's disease and related Lewy body dementia, it's called alpha-synuclein. So several companies developed antibodies, and in mice studies, the antibodies look great. They clear the protein, the mice perform better, but when it's come to the human trials, they've had pretty much mixed results. So we wondered why that was true.
And what did you find?
So what we found is the innate immune system of the cells, of the brain, which are called microglial cells, are obviously not in mice. They have mouse microglial cells. So we took human stem cells and converted them through modern genetic techniques to microglia, just like the human brain has. And what we found is that misfolded proteins irritated those, made them inflame. But amazingly and paradoxically, when we added an antibody to the protein and the two of them bound to the microglia, it made the situation worse. It made more inflammation rather than less. And that wasn't true in mouse microglia. So it's just another example of, unfortunately, the mouse models don't always predict what we're going to see in humans. Now, there is a silver lining to this. It means that those antibody clearing methods might work if we could counteract the inflammation. And we've already developed a drug that we think will work that way. The drug has been in humans before, so we're really encouraged we might be able to convince these companies to use this anti-inflammatory drug in conjunction with their antibodies for both Alzheimer's and Parkinson's, and maybe get a lot more bang for their buck with these antibody therapies.
Yeah, certainly sounds that way. As far as the approach to developing treatments, because as you mentioned, there really aren't a lot or even good ones out there on the market. You are the reason there is one decent one as it stands.
Well, so there are a couple. I'm half the reason.
Okay. Does the approach, you're humble too, I appreciate that. Does the approach at how we try to develop therapies for Alzheimer's disease, does that need to be tweaked a little bit?
Yeah, well, now you've hit my 20-year kind of career in this. What we try to do is find drugs that are already safe for some other indication that get into the brain and that will work on pathways that are aberrantly activated by these neurodegenerative diseases, such as Parkinson's disease and Alzheimer's disease. In fact, we recently started a new medicine center at Scripps Research with my chemistry colleague, Jeff Kelly, for this very, very point. We think we can save 15 years of drug development if we take drugs we already know that are safe and either tweak them, find out exactly how they work in the brain. And the original drug, Namenda, that I developed, was actually developed that way. It was originally an antiviral drug.
Wow, that's so fascinating. Do these drugs behave the same way combined in every person, or is it like a fingerprint and you basically just have to go through trial and error and see what that person responds to?
Yeah, no, that's a great question. We think that there are commonalities, and even commonalities across these diseases. It is true that certain people may have a propensity for one disease or another, but in general, many of the pathways are preserved. And that's why this was such a surprising study, because many of the pathways are preserved in mice. It's just that these particular immune cells in the brain in a human context really react differently than the mouse cells. Wow. Shows you we get the tests in humans.
Yeah, this is fascinating, Dr. Lipton. I really appreciate all the work you're doing, and please keep us updated. This is something that affects so many people, and the research that your teams are doing is changing lives, for sure. I really appreciate it.
Well, I'm also a clinician here in San Diego, so I see a lot of these patients. It's affected my own family, and I'm getting older, as we are all, and we need to work more quickly on all of this.
All right. Well, you certainly are. I appreciate it. Thank you for your time tonight.
Thank you. Thanks.
All right.